This video is to help you figure out how to convert the DNA sequence into the mRNA codons and then into the tRNA anticodons that you're going to use for the protein sentences activity. So the very first thing you should do is draw lines between every 3 DNA bases, because that way it's a lot easier to determine the codons. So like right after ATG, you're going to make a line because ATG is one codon. Then after AAA, you're going to make another line, and that's another codon. And you'll continue that for the rest of the sequence. Next, we're going to convert the DNA to the mRNA. So remember, mRNA has different codons and it's complementary to the DNA strand. And so before we go through how to do those, we're going to really quickly review what bases match up with each other. Remember that DNA has 4 bases, and those bases are A, C, T, and G. And when 2 DNA strands are paired up together in a double helix, A always pairs with T, and C always pairs with G. With mRNA, you've got a slightly different set of codons, right? mRNA has A and C and G, but instead of T, it's replaced with the letter U. So in RNA, you're going to have no T's at all. And so what's going to happen is that we are going to have a complementary mRNA strand that's built from the DNA template strand or the DNA sense strand. And so if you have an A in your DNA, that means that the codon in mRNA is going to be a letter U, because A matches with T normally, but we replace T with U. If you have a T, you are going to have an A. And if you have a G, that's going to match with a C. And so that's our first codon, UAC. Our next codon is going to be AAA. And so if you have an A in the DNA, you're going to write a U in the mRNA strand. And so this is going to be 3 U's in a row. And then we're going to draw a line because that is our next codon. Then we'll do a couple more together here, and then you will be able to work on the rest of the activity by yourself. So again, here we have AAC, which means we're going to have UUG, and then we're going to draw our line. Then if you have AAG, that'll give you UUC. And then we draw our line. And then if you have G, you'll put a C. T pairs with A pairs with U. And we draw our line. So now you should have your mRNA codons done. So we're going to move on to the tRNA, which has anticodons. So anticodons are complementary to the mRNA strand. And they, once again, use the same letters as mRNA. So we're going to use the letters A, C, G, and U. So our anticodon is just the opposite of the mRNA codon. So if you have a U in the codon, the anticodon will have an A. If you have an A, you will write a U. And if you have a C, you will write a G. And then we can do our next anticodon, and that's 3 letter U's. So U will pair with A, so we're going to have A. And then we're going to do our next anticodon, and that's U, U, G. So that's going to give us A, C. And so you'll notice that there are always complementary pairs. You never write the same letter, you always write the complement. And so we'll finish out this strand, and then you can use the codon chart to figure out what the sentence says. So now you're going to take each of those anticodons, you're going to put them in order and build your protein sentence on the document that was either shared with you in Canvas or given to you on paper.